having understanding what is actually happening in other parts of the world. So what is actually happening in our part of the world? That's the question. And this work actually was funded by Plant Health Australia through, at that time, Minister David Littleproud's contribution. He put in quite a few $100,000 to Plant Health Australia to administer the grant, and we were invited to look at the dynamics of populations in Australia, but also through GRDC and again Australian government, looking at the Southeast Asia and the initial population structure and how we can use the information to increase our awareness and our management options. So part of that project through GRDC, CRDC, Corteva, DAO, FMC, and SRA, with ACIR, we then look at bioassays as well, of testing a few chemicals to better understand what is actually happening in Australia. And we actually standardized a protocol across Southeast Asia so that whatever they do and the results is actually interpretable for us in Australia. Because if you read the literature out there, insecticide bioassay is hickety-picklety. Everyone uses their own methods. So at the end of the day, you can't use this because we use it in other methods. You can't use that because we use the other methods. So what we did was we spent a good deal of the time talking to our Southeast Asian partners to standardize the protocol, and I'll show some results. So yes, so we know that you've seen this slide before. Asia has been a hot area in terms of being around this region. So what about Southeast Asia? It's a geographically, culturally, and politically diverse region, and how that is impacting on Australia. So that's what we really wanted to know. So this took place over COVID time, fun time. So every month we have meetings, and it's Zoom or WebEx or Teams. And it's very taxing because there's nothing better than in-person meeting. And the communication difficulties, the expression difficulties is better when you have a face-to-face. So a lot of them, so we have Myanmar at that time before the political development. We have CSIRO, we have Malaysia, we have Laos, we have Cambodia, Vietnam, and we also have Uganda. So this range from the Indo-Pacific region, from around here, all the way up to South Korea. And these are our partner countries, and we also have PNG and we have South Korea. They joined the study at a later stage. So what happened in Australia and in Southeast Asia? It seems like I'm repeating myself a lot. Basically, multiple introduction. Nothing to do with Africa. So what we look at it, there was a paper just published late last year, early this year, and basically went through the paper with this group to basically tell them to look at their genetic data. After 4 reviews, they finally removed a few authors and redo all the analysis, and it came to the conclusion that China, this is their own results, China has all these different colors population representing different introduction events. So if you look at China, you can count at least 6 different events. Okay? In Malaysia, between Kedah State and Penang State and Johor State, we have 3 classes. And interestingly, in Malaysia, in Kedah, this population, when it was first collected, it was brought straight into the lab, and then it had been maintained in the lab. And you can see that when it arrived and being maintained in the lab, it is not anything like the mixed hybrid type population. So the diversity just in this region, just looking at 2 countries, we can at least identify 9 separate introduction events. And taken as a whole, just reviewing the literature and including resistance profiles to different insecticides across the world, so from Asia into Africa, there are at least 20 to 21 events or separate introductions, suggesting that this pest has been coming into our place, into our part of the world, hasn't really been noticed. when you interview the African researchers, you ask the African researchers to go out to interview the farmers, they say that we noticed this type of symptom, this type of pest since 2013 in their village when they're growing maize. We just think that it's a local species. They have no idea, but they have been detecting losses since 2013. But in Uganda, for example, that two surveys were conducted separate time by separate teams, all identified this issue, but Uganda officially reported that it's 2018. So we know that the reporting time, sometimes it's quite politically sensitive, so they delay as long as possible, and it just doesn't help with understanding what's actually happening to this. So this is Southeast Asia, and I'll talk about Australia here. So Australia here, this is the first year when the population first came in. And you can see that Australia really is very diverse, and we're just going to concentrate on here, number 4, which means that to explain the population genetic composition, we can assume, and simulation tells us that if you assume that there are 4 ancestral genetic contributions into the current population, that gives you the best results. So when you look at that, for example, Northern Territory, the pattern in terms of the color is very different from, for example, you have Queensland and also New South Wales. So I'm going to go across to another one. So when we do the PCA analysis, and so if you remember when I said, so we're looking at across Southeast Asia and into Australia. So let's look at Southeast Asia. So if you recall, Myanmar was reporting the fall armyworm by 2018, December. And by 2019, January, China reported it. So as you know, in a strict sense, that's 1 generation, a month. So within a month, you expect the population to be pretty much identical. They're just offspring and they're flying into it. And yet when we look at the genomic pattern, Myanmar and China are completely separate. They are not related. So that brings into doubt how people interpret and use simulation to fit their purpose. Okay? In Malaysia, again, we found that Kedah is here. It's really different from Penang and Johor. Uganda and Malawi, again, they're very different from Asia. In this case, we only have the Chinese population. So it doesn't suggest that they're actually moving in from Africa into Asia. It's just separate introduction. If we look at Australia here, or we look at Southeast Asia. So as a whole, Southeast Asia, we have Laos, Vietnam, Myanmar, Philippines, Malaysia, and Pacific, PNG. They're pretty much, again, very different from China. We're looking at hotspots of biosecurity weaknesses in Southeast Asia and Asia. They are different, and they enable different genetic fitness to be introduced into the current populations. Australia, overall, we have a bit of overlapping with China, but they're also quite different from various Malaysian populations. So, what does it mean in terms of the directionality of movement? When we do the similar gene flow analysis, we can see that from Malaysia, there were bidirectional movements between different Southeast Asian countries. So I'm just highlighting a few. There's quite complex signature up there. And overall, as a simplistic view, at a glance, there are unidirectional movement into Africa, as we already identified from previous studies. But what about coming into Australia? So currently, the assumption is that it came into Australia as a single population. It was detected in Arab and Saibai Island, and then in Bamagat at the top, and then from there, people believe that it spread. So that's what happened, and how do we come to that understanding? Again, we're looking at reverse trajectory studies. So they look at the date when it was first detected in Australia or reported. Remember, these are lures. So you're only really detecting males anyway, right? And you're using female lures, you're detecting males. So if the female came in earlier, you would never see it, right? So in any case, so based on the date and looking at the wind direction, the studies from a Chinese group but involved various Australian collaborators as well, suggested they came in from, I think that is the Indonesian islands of Sulawesi. So it came from there into Sabah and Arab, and then going to Bamaga. It fit the timeline. This is when you detected it, it must be it. But is this the only pathway? And is it really the pathway? Again, we really need to harmonize the genetic evidence with your reverse trajectory. So when we look at that and we survey Australian populations, we have Kununurra, Northern Territory, we have a few of the Northern Queensland populations, and we were from New South Wales. So that's the first year when we had those populations. And what we can see is this little table on the top left-hand corner, is actually the green color means that between populations, so these are populations in a pairwise comparison. The green color indicate that populations were moving. So they share greater similarities of their genetic architect and their genetic signature. And the red is populations are not really moving. So when you look at it at a glance, at a very big picture-wise, Africa, Southeast Asia, Asia, they have reported this pest for much longer, and overall there are some movement, but there are populations that are actually still quite localized. And in Australia, we have some population that seems to be quite cosmopolitan. But overall, in Australia, in the first year, populations were quite localized. And how did they come in? So when we look at this interpretation between India, Malaysia, and Papua New Guinea, we see, if you look at the table carefully, you see that there were multiple pathways. So the pathway link Kununurra populations on the west, more similar to Malaysia, which is again more similar to India. So that's likely one pathway coming in. And then there were very little movements across here. And the populations in, I can't remember the initials, but in Queensland, in this region, were more related to PNG, suggesting that in Australia, you have at least two very separate pathways and nothing to do with it coming from Indonesia going into Queensland. So at least that's what the genetic signature is telling us. And Wewak population was actually really, really different. It's so different, it sits somewhere else. And this is phylogenetic analysis looking at the nuclear markers, so nearly 900 nuclear markers. And what we are seeing is that Australia is in green. You can see that Australia, there's pretty much a big cluster, but somehow we are having Korea somewhere in an ancestral state, suggesting that Korea potentially has something to do with Australian population for some of them. But how did that happen? We don't really know, because Korea to Australia is very far, right? Thousands. I think when I last looked at it, I think if I remember correctly, about 7,500 kilometers. So yes, 160 kilometers you can fly at night, but 7,000 kilometers, come on. So we are not too sure how that actually happens. So Australia is there, China, basal to everything else. There's some African ones sitting somewhere else as well. And what we're seeing, and it's hard to see, but there's this black line here. That's the African population. It's sitting within American population. So how did that, what does that mean? It potentially means that these individuals with the genetic material originating from Africa have been introduced back into South America, potentially, right? So these are the signature, and we can see that Australian population sits everywhere else across Southeast Asia. So we're having different contributions from different Asian and Southeast Asian populations. And interestingly, what we see is that African population, East African population, sits at the edge, at the tip of Asian populations, suggesting that they originated from Southeast Asia. So again, the movement is completely opposite, and we have a really, really hot biosecurity weakness in Asia and in Southeast Asia. And is this potentially an accidental introduction through trade into Australia? that remain to be seen. But it does make you wonder why we have such diversity, basically because there is a lot of weaknesses in our global biosecurity protocol. So in the second year and third year, we look at the movement of the 4 armyworms within Australia. So the red dots in Australia represents populations that we look at previous year, and we use the same approach to include new populations that are represented in the blue dot. And unfortunately, again, it was over COVID time, we lost the Western Australian population, so we really have no idea what happened in the second year and third year for Western Australia, but we have contributions from Melina, from Kevin, and from New Zealand, refugees, to look at what's actually happening. So I really appreciate all their input, and I want to acknowledge them here publicly. So this is Australia, second year and third year. Compare with first year in red, second year, across year, and this is New South Wales population. And this is, in this case, this simulation looking at the population clustering suggests that at K, or genetic cluster, that's what it represents, of 5 genetic clusters, it explains your population the best. And when you look at New South Wales, you look at how different the signature is, especially all these black individuals. It's nowhere found in anywhere else in Australia. So that's really quite surprising. In Northern Territory, predominantly we have signature that look fairly homogeneous, and it's a bit similar to previously in Burdekin. These are Burdekin, Queensland populations. They're a bit similar, a bit, not identical. But then when you look at in the current year Queensland population, they're quite different. So we're looking at very uneven distribution of these pests. And this has implications because if the pests were, as suggested, it came first into northern Queensland and then spread everywhere, we can develop a national management policies very quickly. Everyone uses insecticide for this year, this time, this crop. But we're actually looking at really heterogeneous genetic composition. And that needs to be taken into consideration when people like yourself and other industry partners are sitting down and trying to develop a management strategy. You really need to consider how different these populations are. And this is just a snapshot. They are continuously coming in. New Zealand, we only have 4 individuals, so we can't really say a lot other than to confirm that the fact that they are actually hybrid as well. So we have a big clusters of Australian populations, but we have really quite unique groups coming out. And these are analyzed at the same time, and they just don't fit in your current scenario of a big homogeneous populations. How these populations respond to Australian climate, to the crop landscape, we don't know. And those are the things that we require assistance and collaborations with you guys to further understand. So I just want to very quickly summarize the genomic component of it before I move on to resistance and bioassays. So obviously multiple introduction I've mentioned multiple times. We don't say that just lightly. We look at whole genome analysis, and we see that at global scale, Asia, Southeast Asia, really they're biosecurity hotspot, and they're just our neighbors. So Australian government, we can invest $400 million every year to protect our borders, but if we don't help our neighbors, at the end of the day, that money is going to be wasted because whatever they have, we will get. Especially if a lot of them we believe are natural migration. You can't stop it. You need to help them to better their biosecurity preparedness and detection. Stop them, we help ourselves. And from that, DFAT is currently funding a project that we are looking at developing an ASEAN Bioprotection Research Center. And I'm leading that project. And that's to basically, with a colleague in Singapore, Alison Watson. So Alison's doing most of the work because I'm just badly organized with time. So we are looking at partnering ASEAN partners to there's a general wish to use insecticide rather than harsh chemicals to control this pest. So we're trying to help them to develop that and to help them to utilize genomics, as well as there's a gender issue as well, because in Asia a lot of the farmers are females, but because of the cultural issue, females' opinions are generally not considered important, and they can't even get funding to manage a farm unless their male partner say so. So we're trying to help them to overcome that. So that ASEAN Bioprotection Center has a very important gender aspect as well. So the spread pattern, really we need to reassess it, and could collaboration between global biosecurity agencies have helped us manage and better protect us from this pest? And this is important because other pests are coming. I'll show you a bit later. So if you go to USDA website, Lab Intercept, and you look at their interception data, that website was last updated before 2014. They list all the countries that they intercepted fall armyworms coming back into USA. Thailand, India, Israel. The list goes on. China. So yes, this pest, I believe, has been in our region for a long time, and we haven't really paid attention to it. We're quite poor at understanding. Department of Agriculture, 2013, we actually intercepted or reported to have intercepted material coming from Vietnam before 2013. So that's amazing. And then when I mentioned to our Vietnam collaborators in this project, they said, oh, we got a thesis. 2008, lawn across Hanoi parks were damaged. We measured their damage level over multiple months. 2008. And because they do not know what to call it, they call it differently. Nobody pay attention to it. And that's published. But in Vietnamese, and I don't read Vietnamese because I'm not Vietnamese. So, yeah, and we really need to approach this with a whole genome analysis, and we need to understand the limitations and the research providers and various scientists that are using molecular markers. My advice is don't push and overuse your marker. They are for something, and you just use it for something, for that purpose. If you use it to infer other things, you could mislead and complicate the situation. So, okay, what about, who cares if they have multiple introductions? We should care. So, Spodoptera fujiperta has resistance to all these chemicals in North and South America. So Cry1F, fall armyworm is extremely sensitive to Cry1F. The maize GM line was lost in Puerto Rico because they developed Cry1F resistance. So that technology is lost. So should Australia go for GM maize? What type of preparedness do we have in terms of industry understanding and industry willingness to uptake it? Is there any appetite for it? I'm not going to promote it or dissuade it. I'm just here. If you want to discuss, we can try to discuss it. But there are different resistance. VIP3A, really important, but there's resistance. And VIP3A is also confirmed in, since this site was prepared a while back in Brazil. So VIP3A is really important again, because in Australia we have VIP3 or Bogart 3 cotton targeting Punctigera and Armigera. So we know that in Australia there are VIP3 alleles out there in fall armyworm, in Helicobacter Armigera. But what if fall armyworm came over with a VIP3 resistant allele? Well, in a little while you see that fall armyworm doesn't really care about Cry1AC. It just doesn't affect it. It's slightly impacted by Cry2AB, but it's very sensitive to VIP3. So for Australian cotton industry, the technology that's still being used is Bogart 3, has these 3 toxins in it. If for armyworm with a resistance coming across, not effective, maybe, maybe not, and don't care. So you lost your technology immediately. So that's really, really important. Knowing the pathway protects our industry. It's really, really important. And the global pictures of resistances are so diverse. We are constantly discovering new subspecies. Subspecies in the sense that at the genome level, they truly are very different. They're still considered as a Helicobacter armigera in this case. But in Xinjiang, for example, they're extremely tolerant to cold. And you look at their genomic signature, they're separated from the rest of the armigera about 200,000 years ago. Armigera and converter, another subspecies for Australia and New Zealand, separated about 100,000 years ago. So we have a lot of population diversity with different capability of fighting back what we're trying to control them. So it's really important to get the biosecurity right. So these are the chemicals that we're testing, or we've tested in Australian populations. And we try to get the Southeast Asian partners to test some of these because they have limitations in, first of all, rearing the colonies that they're using, and the time is very short. And then there are difficulties in accessing the different chemicals. So they do what they can. And these are the chemistry that we tested. So that in cooperation, we have chlorantraniliprole, indoxacarb, amamectin, and spinetoram. Topical application, we have cypermethrin and metamol. And then we do surface treatments, BT, VIP. So we look at Cry1A, Cry1AC, Cry2AB, VIP, Cry1F, and Zentari and Dipel. These are commercial products. Yeah? And this is published, by the way, everyone. So you can read the paper. Of course, no problem. And we do it with Australian populations. In essence, we do the workman population, which was supposed to be the ground zero population, the first population that was detected by University of Queensland. They sent it down to us. And then we also got populations from Kununurra. So we are testing 2 different populations in Australia. We do it at CSIRO. And we have the various different countries from Asia doing the other bioassays. So we have published this report, we submitted this report, this is done, this is how Bill, before he retired, he essentially helped set up the 4 armyworm colonies, and he did just about all the bioassay, amazing person. So we have Tom and Serena, again, researchers, senior research and principal researchers just working on the 4 armyworm. So Serena now has a broad acreage project by, I think it's by Hort, or GRDC, I think, and looking at management, IPM management, and 4 armyworm might be a consideration as well. So it's really important because publications like this from, for example, Philippines, they say basically using the limited genetic markers of CO1, it's all homogeneous. And given that for the signature look the same as India and South Africa, it must be coming from Africa. And it's given that there's no Cry1F resistance in Africa, it must be the same in Philippines. And Philippines has BT maize. They have benefited a lot from using BT maize. So when I was in Philippines just late last year, they said, no, 4 armyworm, not really an issue on our maize. On our rice, yes, it's being attacked. But on the maize, no, because we use BT. So by assuming that Togo has no resistance, so they can be quite safe, they're actually forgetting that if the pest did come in elsewhere, carrying a Cry1F resistance, there goes all the investment. Right? So looking towards Africa is not acceptable. The idea that resistance evolved really quickly in 4 armyworms. So this study came out and is led by Ralph Nauen. A lot of you might know Ralph. Bayer's chief scientist and in insecticide and all that. So really great studies. They look at multiple populations between years, multiple sampling times. And as a whole, if you look at, like 2 years, so 24 generations approximately, there's really not a lot of changes in these populations. They're quite static. There are a few populations that are a bit outlier, for example. Are they representative of rapid evolution of resistance? We don't know. Or are they new incursions coming in with new resistance? Again, we don't know. They didn't test it. They didn't do genomics. They just do bioassays. So it'd be really, really good to really understand what's actually happening. But for Australia, we really need to do something like this. Are we seeing rapid evolution? When you're using certain insecticide, are you actually selecting for it? Do we have similar scenario in India, for example? We actually do not know a lot. And there's a lot of values in the next few slides, so I'm just going to highlight the differences. So, methanol. We do bioassays, so we have SF20, so the year 20, and the 1 is the first population we have. So Queensland, that's the founder population in Australia supposedly, and Western Australia. And if you look at methanol, really it's not much different. And South Africa methanol, huge resistance. And when you compare that with, that's when you compare with what's considered as US, Florida susceptible population, it is really, really high. But if you compare it with the US, Florida resistant population is still high. It's really quite different. So if I have a choice, I don't want this population to be coming to Australia. We want to be monitoring for it. We don't want this population. And if we look at, for example, the other bioassay that we did for the project that was funded by GRDC and ACRR with all the chemical partners, we look at just a few, again, methanol, alpha-cypermethrin, and in here we have Indonesia, Laos as well, and we do it in Malaysia as well, and we do it against Armyworm because Armyworm is the pest that most of you are familiar with managing it. So we compare that in terms of toxicity ratio against Armyworm, just to understand what the hell is going on. And you look at Laos population, really very high. You don't want Laos population to be coming to Australia either, right? Okay, again, Australia population. Western Australia, 52 times more resistant than Armyworm as compared to Queensland. And perhaps, Subra, that's the reason why you are seeing different responses, because as we show by the genomic analysis, there are really, really different pathways coming into Australia. They do not just spread into what you currently have. So how do we manage something like this as a national policy? I don't know. A lot of work. And okay, some Southeast Asian samples and comparing with, in this case, BT. So BT being a biopesticides, this is interesting. So BT, so as I mentioned before, BT, Armyworm in Australia, they're highly sensitive. In Asia and Africa, for example, they're resistant out there. But in Australia, we didn't have Cry1AC resistance. But when you look at for the Spodoptera, so we at that time our Litura population was still going, they don't care because it's 1AC. It is ineffective against Spodoptera. So in that sense, there is implication for the cotton industry, for example. And can you use a Cry1AC from the market to control your fall armyworm? You probably won't, because we are showing it that it is ineffective. It is, as literature said, 1AC is ineffective against fall armyworm. 2AB, just a little bit. Yes and no, it all depends. VIP3, when you compare that, is highly sensitive. And when you look at Dipel and Centauri, which I think in the previous slide and and also in the literature that we publish, we show that you can work out what they actually have, even though they don't specifically say that they target fall armyworms. And you can see that, for example, Centauri is quite effective as compared to, for example, Dipel. Okay? So for organic farmers, perhaps you might consider Xanthari, but I'm not being paid by them. I don't know. You do whatever you want. Okay? I hope that makes sense. And then in terms of Southeast Asia versus Australia, you can see that despite the fact that 1AC is not really sensitive for armyworm in general, but the population in Indonesia, the one that our partner tried it out, it was highly sensitive, which is really surprising. And Cry1F in general is very sensitive to all the 4 armyworms. So if you buy your product and it says Cry1F, you probably could use it to spray, but you hope that there's no resistance evolving or resistant allele coming in. And spinosad, diamide, organophosphate, we don't really use it anymore. But we can see that at the end of the day, when we compare that with all the publications, there are really quite different pathways that we can say that these populations are unrelated, they are independent, and you don't want to have these populations coming into Australia at the end of the day. So we need to continuously monitoring, and we want to help our neighbor so that they don't have new populations coming into them. And finally, we are looking at, say, in this case, chlorantraniliprole, indoxacarb, and amamectin. So in this case, I highlighted the blue and the red because when I was in Malaysia, they say their insecticide resistant management protocol is to rotate between chlorantraniliprole and amamectin. So I would say that that's probably not going to work very long because they're starting to see high resistant frequencies in amamectin. So when you rotate it, you really want 2 that are quite sensitive. You don't want that that's already on its way out. So again, how we actually implement our resistant management plan will depend on what we are facing, and we need this type of studies, bioassay studies, on a standard protocol. And again, indoxacarb, for example, in SF20-1 in the Burdekin Walkerman population, it's very low, but in the Western Australia population, it's huge. And this is 2 months apart, 2 generations. There is no way your resistance evolves that fast. Okay? So, have we learned our lesson? This is, I promise, it's the end. So we are supposed to be feeding 10 billion people by 2050. We are here now. We're 8 billion, 2023. Just facing these 2 pests alone, Armyworm and Spodoptera, is going to be really, really difficult. And we have all these other related major pest species dotted in all different areas of the world. And as you can see, we're really, really poor at doing our biosecurity. Despite we say that we're really good for Australia, we're still quite poor. We don't help our neighbors enough. And when we look at the interception data, Armyworm is here, Spodoptera is here, and that's Fujiperta. It overtook Armyworm. Okay? And we have a lot of samples. In this case, it's a combination of various samples that we actually have no idea what they are. The diversity out there has no genetic signature that we can trace back. So how do we do proper biosecurity? So what? Well, we actually have Litura, and you can see that Litura, through our interception data, we intercepted it coming back from here into Australia. This is a species native range being in Asia, and yet we're detecting them coming back from Americas. They have already established population there. So when it explodes, probably climatic factor, trigger point, who knows? So that's really quite concerning. And if you look at the phylogeny of, this is the latest phylogeny of Spodoptera genus, all the red arrows are known major pest species dotted around the world. We're only looking at here, 1. What about the rest? And this is important because take fall armyworm as an example, asparagus and cut flowers only. You can find this data out there in the internet. You can look at your export, import values in US dollars and all that. Previously, so in fresh vegetable, for example, we probably only concerned that fall armyworm will be coming from here, and that's it. Nowhere else, right? But, so we do have pressure, but nothing in comparison when we actually have all the other countries having the pests trying to come into Australia, right? So that is really important. We need to help other people. And does it matter? So this is Littoralis, native range is here. In our interception, so Littoralis is probably about 20 or 18, something like that, top 18. Anyway, currently if you go to Apple website, this is the spread of Littoralis. But in our interception data, we can see, and in other websites, you can see that people have been reporting it. 1999, 2000, 2015, 2022. You think that it's gradually being detected across different regions into Southeast Asia. There was even one supposedly reported in Australia. Whether this is true or not, we don't know. They don't provide enough information. So this is another pest that is actually on the move. It's similar to Spodoptera frugiperta. These are the pests, food crop, 44 families. And it's called the Egyptian cotton moth. So that's for a reason. It attacks cotton. And 8 families of the crop that it attacks is more than 50% of the host in the family sense. And it attacks all this. So just as a parting advice is, we probably should start monitoring for these pests, to be honest. So as we're still working on fall armyworms, CSIRO is actively trying to establish collaboration with France because this is a species that is in Europe and CSIRO has laboratory in France. So we're going to try to get the France colleague to start understanding the species. But again, we need funding. So I'm in the process of trying to negotiate for similar funding through CSIRO to try to set up this program. But yeah, this is not the only one. There are all the other red ones coming in. So with that, thank you. I want to acknowledge everyone up there for all their help, especially the funding agencies and everyone in the room and online. And thank you.